I'm SFI Tarnita. I'm Princeton in ecology and evolutionary biology. My training is as a mathematician, but I work now to try to understand the organization of complex systems, and we ask both evolutionary and ecological questions, and we look across a variety of scales from cells to ecosystems, trying to extract some general rules. And today I'm going to talk about one aspect of the work that we do, and that's the origin of biological construction. So if we think about the way that nature constructs new things, it seems to always be, and by new things, and I would particularly focus here on hierarchical construction, where you take units, somehow they are put together to form a bigger entity, and those entities can also be put together to form bigger entities, and so on and so forth. So cells forming multicellular organisms, or animals forming animal societies. If you ask, what are the rules, what are the mechanisms employed to construct these new complexes, there seem to always be two, or some combination of the two. You either start with one individual, be it a cell or an ant, and when it reproduces or it divides, the daughter cells don't separate, they stay together, and they continue like this, and they build something bigger. Or you have completely independent individuals that for some reason, at some point in their life, they come together, perhaps transiently, to solve a particular problem. Let's say they have to weather starvation or predation or some such thing. So there are two. I mean, in some sense, from a mathematician's point of view, this is beautiful, right? Pretty much every single instance that has led to a construction event has followed one of these two. So there's hope to understand. I mean, in this simplicity, there's hope to understand something about this diversity of life. There are, however, three big questions that emerge as you start to think about this. One is, how do you actually get diversity? If the rules are so simple, and you always start with one unit, how do you get a diversity of life cycles? How do you get all these different multicellular forms that reproduce in different ways, and or social groups that have very different types of organization? How does complexity arise? You start from one thing that divides, you get a bunch of things that are all the same. It's pretty boring. How does that lead to anything more interesting and more complicated than that? And then you can ask questions about what threatens construction. What is it weak to? What are its weaknesses, and what makes it robust? And actually, the very simple way of setting this up has allowed us, these very simple way of looking at the world has actually allowed us to answer a lot of these questions. Amazingly, many of the answers are actually universal in the sense that they apply to ants just as much as they apply to multicellular organisms. But at the same time, there's still actually a lot unknown, especially when it comes to the first two. And in principle, our theoretical approach for answering these first two questions has been to say, it's kind of a top-down approach. We start with something that we think is amazing, usually human beings. And then we say, what characteristics make them amazing? And then we ask, how can something like this lead to the organization of a human being? And typically, the answer has also been kind of step-by-step and sequential. We start with something boring. We have a group of boring cells. They have to cooperate. If they cooperate really, really, really hard, one day they might start to do different things. And if they start to do different things, they depend on each other, and there we have it. We have some sort of division of labor or differentiation. And if that happens enough, we might get some spatial pattern formation. So there's kind of a sequential way of building the complexity. But actually, I think a lot of that perspective, while it may seem intuitive, has actually been kind of flawed. So really recent work in comparative genomics, very cool work, has shown that, for example, if you look at the evolution of multicellularity, a lot of the innovations that we thought were characteristic of multicellularity, only multicellular organisms could have invented this, were actually things that you could find in the ancestor. About 80% of the functions that we thought were invented by multicellularity, you can find them in the solitary ancestor. And so they perhaps were doing different things, and they were co-opted to do something novel within the multicellular organism, but they were there. So then, to me, this leads to a more interesting, first of all, it leads to a framework, a theoretical framework that requires us to build the ancestor in there in a more interesting way. And so I'm going to give two examples of what we've learned recently by doing such theoretical work. So I started to think about these kinds of questions with the evolution of eusociality. Eusociality is this amazing social behavior that happens that ants and termites and bees have. The most important ingredient of it is that it needs to have division of reproduction. So certain things must divide and others must reproduce and others must not ever. How does that evolve? Well, we proposed a very simple model. The point of this part of the talk is not actually to go into the depths of the model, but it is a staying together model, like the one, like the very first mechanism that I showed you there. You can start with a solitary individual that lays an egg here, provisions it with some food, moves around, lays another egg, moves around, and so on and so forth. And contrast that, or compete that, because they're competing for the same resources in the same environment, with an individual that's able to produce some daughters that now stay at the nest. So some fraction of the daughters of this individual now are able to stay at the nest and construct bigger groups, whereas others are going to go off and start their own colonies. When would this ever be favored? The answer is yes, but it's very hard to do, and a very important ingredient for it is that it requires for the benefits of being part of this sort of lifestyle to kick in at very small group sizes. So small that I actually really thought that there was something wrong with the model, because no matter how I looked at it, it had to be, you had to get benefits at less than 10 ants. Okay, at the very origin of eusociality, those 10 ants would have been, or wasps, would have been so similar to each other that it really, I found it very, counterintuitive that what kind of dramatic event would have to happen to give an enormous fitness boost to a group of such a small size of basically identical individuals. Well, I stopped thinking about this a decade ago and revisited it just a couple of years ago, because finally some colleagues at Rockefeller were able to rear in the lab ants that were clonal. So they could control everything about their, we could make them be genetically identical, have the same age, have the same, more or less the same morphology, meaning that they have the same size. So make them as similar as possible and try to replicate as much as possible the conditions at the origin of eusociality. And then try to form groups of different sizes with these ants and see what happens to the fitness of the colony. And they were able to do this with groups of size 2, 4, 6, up to 16, and track every single individual in the colony. And this is what happened to fitness. Not much happens when you have groups of size 2 or 4. 1 is really bad, but it can persist a little bit. But once you get to groups of size 6, you start to get really close to a doubling of colony. And that plateaus at basically size 16. And we know that this is exactly the growth of a colony like this that has 500,000 individuals. Okay? So with 6 ants, they're able to, 6 to 8 ants, they're actually able to be almost as good as if they had 500,000. We did some modeling and we worked with them with the experiments to basically show that what was actually happening in there was rudimentary specialization. But so rudimentary that none of the definitions of specialization or metrics of specialization that we would use to look at, cell differentiation, to assess the amount of cell differentiation in a human being or in a super sophisticated ant colony would have actually shown us that there is specialization. So we basically had to come up with a different metric to see what these ants were actually doing. And if you're curious what they were, you have to ask me at the end, because I want to say one other small story. But in principle, the bottom line was that synergistic interactions, even when the units are identical and really, really few, can still harness the behavioral repertoire of the ancestor to lead to emergent phenomena, basically spontaneously almost. Now, very quickly, a story about how can you get diversity, again, without requiring something very complicated or a lot of evolution to happen. And here I want to talk about the origin of diverse multicellular life cycles. We have typically, again, when we think about in all of these multi-level selection theory kind of approaches or kin selection, what we care about is a very boring ancestor. All we care about it is that it's very cooperative and very related to something or other. But we don't really think a lot about what that ancestor was actually doing. But what if we actually consider a little bit, think about it a little bit more, and think about some of the possible ancestral constraints and ecological conditions that existed. So what we did was to start with an ancestor that was equipped with a gene regulatory network. So it had the ability to actually evolve in response to the environment. We didn't give it a repertoire of strategies. We let it discover those strategies. And we give it an environment that was just a little bit more interesting than nothing happening. Two things were happening, and it was completely predictable. Alternating environments, let's say seasonal or diurnal, but they had the same length and periodicity and so on and so forth. And then we said, okay, well, what this ancestor has to do is it has to, in this environment, the best thing for its fitness is to express gene A in environment 1 and gene B in environment 2. That's it. We also said that perhaps the ecological conditions might change a little bit at some point in environment 1, so that promiscuously, so this is not a selected function, but promiscuously, when you express gene A, that cell becomes sticky, so it can form groups. Then we have exactly the story that I showed you earlier with the ants, the exact same model, because again, this is a universal way of building it. So it's a staying together kind of model. They divide, they don't separate. And we give them some ecological costs and benefits. Perhaps if they're big enough, they can escape predation, but there are also some costs, because if you're part of a group, you might share resources with other individuals in the group. There's now a potential tension between the selection on the ancestral function. The ancestor still has to do the same thing. It still has to turn on gene A in environment 1, still has to turn on gene B in environment 2. But sometimes it might make sense to form groups even in environment 2, because you get really high benefits from escaping predation. So there's a tension there between what the ancestor had to do and the potential costs and benefits that one gets now out of being multicellular. But the only point to say so far is that this is not too complicated. All we did was to just say the ancestor can be just a little bit more interesting than a circle. If we do that and we allow selection to happen, very quickly we find actually a diversity of life cycles. And here the life cycle, I'll just draw attention to one of them, perhaps my favorite one, because I never would have thought to actually put this as a strategy into the model, so it was really revealed by the existence of the gene regulatory network, is one that does the following thing. These are the environment, 1, 2, 1, 2, and it cycles. This strategy is to have gene A turned on the entire time, except at the transition between environment 2 and environment 1. So this is a multicellular organism that grows, and all of a sudden, poof, it just separates into all of its cells, and then they rebuild the whole thing again in the immediate next step. This is basically like slime molds. So we didn't set out to actually model something very complicated about extant multicellular life cycles, but in this very simple setting, we recovered a diversity that is also biologically meaningful. This is also something that looks like animals, for example. You always form groups. Every once in a while, you have a single cell that separates. If I also give them the other mechanism of coming together, then I find even more life cycles. So we have 6 of them. That's it. And so, the bottom line from this second part, or actually the whole part, is that a theoretical framework that integrates the ancestor a little bit more with its behavioral repertoire and its constraints and its ecology might actually give us a lot of interesting results about what's possible in terms of diversity and what's possible in terms of complexity, and even guide us to better think of what complexity might even be, because a lot can come out for free from what the ancestor was already able to do. All right, that's all I have. I don't know, I just think it's Doug now, or?
Well, you can ask questions, yes, while Doug is coming up, yes.
All right.
Can I just ask Bob's question? It felt a little bit along the line of what people do in reaction to fusion, right? Which is there's a sort of a space of parameter values, and under different parameter values, you can produce all these patterns. So I'm just wondering, I want to ask the robustness question, because you sort of alluded to that as your third principle. So you can produce the diversity, I don't know about complex, maybe, but what about the robustness issue? Do you go from being a slime mold to a leopard with a change in a real value? Yes, with by 10 to the minus 6, you know.
Why is that a robustness question? Is a leopard more robust than a
No,
slime mold?
I'm simply saying, can you move between life cycles with small changes in your parameter values?
Ah, that's an interesting question. I mean, these were all evolved in very different regions of the parameter space. We never really thought, you know, can this one turn into something else? We haven't really asked that question, but that's actually an interesting question. I hadn't thought about that. I think to me, the more interesting aspect of it is Doug and I were talking about this. Often we talk about complexity as, you know, we define a complex thing to have, from the perspective of multicellularity at least, you know, to have a lot of differentiated cell types. But what is a lot? Where do we draw the line? In fact, if I make a very similar model, very simple model with an ancestor that's able to do, you know, 5 different things, you're going to get a multicellular organism that can spontaneously have cell differentiation into 5 different things, and do that very meaningfully and very interestingly, right? And so is that kind of challenges the concept of you're more complex somehow, or you've evolved to be more complex, right? So that was one of them. Yeah.
But empirically, the answer would be yes. So if you look at Nicole King's choanoflagellates, you've got populations that can vary between completely unicellular and multicellular forms. So
you can have that, but I'm not sure if you can change life cycle entirely, you know, between
But that does affect our life cycle.
Sure.
Yeah.